Hello there. As we go into e-learning, um, our first goal for this first week and a half was supposed to be to go over cell parts and functions, and then as groups to develop a factory that relates to cell parts and their functions. So obviously we're going to modify that. What we do exactly with the cell factory or the factory you're going to develop will depend on when we get back into school. So what I'm going to do, I'm going to save you having to read the book. You are welcome. And I'm going to go through the cell parts and their functions. So I know in Bio 1 you went over this, so I'm not going to spend a ton of time on it. Once I go over this, the responsibility is for you to go over these parts and their functions. Um, and we're going to try to apply that to a factory. Okay. All right. Let me get to the next slide. Okay. So what I have here is something I was going to give you anyways, and I will give it to you when we get back into school. Um, is just a picture with the basic cell parts and functions. So I'm going to go through them. I would recommend that you take notes over what I am saying because it's in my terminology, which is what you will see on a test or what my expectations are with that. Okay, so you're going to build another type of factory, but basically what a cell is, it's a protein factory. And the other day you went over different proteins in the cell membrane and their functions, and that may have been new to you. But basically what cells do is they make proteins. Proteins are the most diverse molecule. We talked about these last semester, and what your cells are doing is making them, okay, is making them. And then there are other organelles whose job it is to support that making and that production of the proteins. Okay. So these red terms over here, they are just additional and trying to simplify some of the parts and what they do. So I will go over there as we go through. These I've crossed out. So just remember when you look at the sheet I'm going to give you, not to worry about those. So we're going to start with the nucleus. The simplest definition of the nucleus is it contains the genetic instructions. So every protein in your body, we'll get into this with the genetics, and you should already kind of know this. Every protein in your body has instructions. Those instructions are within the nucleus, specifically within the chromatin, within the DNA. Within the DNA. So the nucleus is where those genetic instructions are located. They need to be protected. You don't want anything bad to happen to them, so you want to put them somewhere safe. The nucleolus is a structure within the nucleus where ribosomes are going to form. Okay. So what's basically going to happen in this factory is your instructions, your blueprints for the proteins. We'll talk about this with, you guys might remember this from Bio 1, protein synthesis, transcription, translation. Hopefully that rings a bell. But you'll make copies of these instructions for the proteins. They will go to the ribosomes. The ribosomes are where the instructions for the proteins, where the proteins will be made. So to synthesize means to make. So it is where the proteins, based on the instructions, where the proteins will be put together. Where the proteins will be put together. Then you have the rough and smooth endoplasmic reticulum. In general, what the endoplasmic reticulum does, the rough is it shapes proteins. So the rough endoplasmic reticulum is called the rough endoplasmic reticulum because it has ribosomes on it. That's what makes it rough. So basically what happens is once the protein is made, remember proteins are made of amino acids, once that strand of amino acids is made, the ribosome, I always use the word poops, but it puts that protein into the rough endoplasmic reticulum, and then what it does is it's going to shape and modify the proteins. Okay. Smooth endoplasmic reticulum. Smooth endoplasmic reticulum is smooth because it does not have ribosomes, and what it's going to do, it's going to synthesize lipids. So it's going to synthesize something besides the main product of the cell. The main product is proteins, but the smooth is going to synthesize lipids. Once it's in there, the proteins are in there in the rough, it's going to shape and modify them, and then it's going to head to the Golgi apparatus, which is going to then modify and ship the proteins. So when we get back to school, I will show you the cell model. You may remember it from Bio 1, but I will show you the parts on the cell model that we have at school. So it's going to modify, and just as importantly, it's going to ship the protein. Now, where can it ship it? Well, if it's a protein for the cell to use, then it's going to ship it within the cell itself. So think of the channel proteins. It's going to take the channel protein that has been made and ship it to the cell membrane and put it on the cell membrane. If it's a protein that the body needs, let's say it's a pancreas cell, I think that was in your video, making insulin, then it's going to take the Golgi apparatus is going to ship it to the cell membrane, and then through exocytosis, it's going to release that protein into the bloodstream, where it will then go and then do its job. So again, it can ship proteins within the cell, it can ship proteins outside the cell. Okay, so that is the basic idea and the basic structure of how proteins will travel throughout. What's going to help with that? Well, you have to have energy for that. Okay, you have to have energy for that. So that energy is going to come from the mitochondria. So when I taught at VU, I would have students in their 40s and 50s who hadn't been to school since high school. And the one structure that everyone remembers the function is the mitochondria. So it forms ATP through cellular respiration. We're going to go through that after we get done with the cell, and then we're going to do a little writing project, and after that we'll go through that in detail. Okay. You also have the peroxisomes, which are going to metabolize waste, and peroxisomes and lysosomes. Lysosomes are going to digest food, so in order for the mitochondria to make energy, it needs nutrients to do that. And so the lysosome can digest that and then provide the nutrients to the mitochondria. It can also get rid of waste materials. Peroxisome is going to metabolize waste products. So an example of where you would see this is in liver cells. So all your blood goes through your liver at some point, and the liver cells are loaded with peroxisomes. And what they will do is, let's say someone drinks alcohol or takes a drug. As the blood goes through your liver, the peroxisomes will break down the alcohol or break down the medication or break down the drugs to then decrease your blood alcohol content and so on. So someone who drinks a lot of alcohol, takes a lot of drugs, a lot of times their liver can kind of just wear out because it's been metabolizing and is exposed to all these waste products and all these drug products throughout a lifetime. So peroxisomes, lysosomes. Okay. Other structures you need to know are the centrosome and the centriole. I'll show you the difference in structure when we look at the model, but the centrosome, okay, the centrioles are within the centrosome. So on the model, the centrioles are yellow, and they're little proteins, and they are located in a black structure called the centrosome. The centrioles, and again you may remember this from Bio 1, are going to assist with the process of mitosis. So when a cell goes through mitosis, it divides into two. These are going to help with that process. Microtubules. They are going to maintain mitotic spindles, which is going to again deal with mitosis, and they're also going to give the cell unique shapes. Okay, so I'll show you a neuron. A lot of cells, muscle cells do not look like the cell model that we use. They look very different. So very unique shapes come from microtubules. Microtubules, and you can add this to your notes, also things can be transported along microtubules. So you can think of it like a railroad track, but it's a railroad track that's not permanent. So what happens is if a protein needs to move from the endoplasmic reticulum to the Golgi apparatus, microtubules can be put down, the protein can travel along there. Now the nice thing about this is when the protein's done traveling, then the railroad track comes up. Unlike the railroad tracks in Vincennes that never go away, these microtubules can do that. So add that to your notes. So we haven't really talked about the nuclear envelope, but it's kind of like the cell membrane, but around the nucleus. We talked about chromatin, talked about nucleolus, is where ribosomes are made. Peroxisomes, we went through the rough and smooth endoplasmic reticulum. Cytoskeleton, centrosome, lysosome, Golgi apparatus. Cytoplasm is just everything within the cell membrane. I'll never ask you that definition. Mitochondria we've talked about. Vacuoles store water. They're pretty insignificant in animal cells, but they do store water. And that leads us to the next part of this that you need to kind of know. This won't apply to the factory assignment, but the differences between plant and animal cells. You'll have this handout, so you can just look at that. So what I'm going to want you to do for the first day, for Tuesday, is go back and look at what I wrote. I don't want to put a set assignment in here because I may use this video other times. So look at what the assignment is. It's basically going to be note taking, and then we will go from there. So if you're having any issues or problems or just are bored, feel free to send me something on the Remind app. I've spent most of my extra time through the snow watching the birds and feeding the birds and watching the cats watch the birds. So I'm ready to go back to school. So I hope you're all doing well, and I will see you soon.